Hello, my name is Adara Bacon, and I'm working to synthesize novel disulfide-linked peptide nucleic acid monomers. Peptide nucleic acids, known as PNAs, are synthesized polymers that are similar to the RNA and DNA, allowing them to bind, hybridize the fancy word we use, to the complementary strands of DNA and RNA to form duplexes. As PNAs do not contain a charged phosphate group like DNA and RNA in their backbone, duplexes formed between DNA and PNA and RNA are thermally more stable than DNA RNA or DNA DNA duplexes, as there is no electrostatic repulsion present. Moreover, since PNAs don't look exactly like DNA or exactly like a protein, they're not degraded by enzymes, specifically nucleases and proteases that typically destroy the bond molecules. However, due to PNAs lacking the charged phosphate group and polarity in the backbone, PNAs are not water soluble. Therefore, they cannot easily penetrate the cell membrane and decrease their ability to be utilized in applications inside of the cell. To combat this issue, we propose developing a PNA duplex with a temporary delivery PNA strand held together by disulfide linkages, which are just two sulfur atoms that share electrons to create a covalent bond. This temporary strand is covered by chemical markers and proteins that are able to penetrate the cell membrane and ultimately trick the cell into taking the PNA duplex across its membrane. Once inside the cell, the disulfide linkages of the temporary strand will be broken down by glutathione, which is an antioxidant made inside of our cells at high concentrations. For my research specifically, I am focused on synthesizing these novel disulfide-linked monomers that will be incorporated into the backbone of the PNAs. The PNA monomers that we desire contain modifications near the disulfide bond. To synthesize them, we perform a phase certification where water is a secondary product along with the intermediate compound B. We can exploit this to drive the reaction forward by removing the water, causing the equilibrium to shift to the right towards our desired product. We use this intermediate to synthesize compound D through a disulfide exchange, where two symmetrical disulfides in the reaction will switch partners to produce asymmetrical compounds. While we have successfully performed this reaction, it is far from being optimized, which will be a part of our future work. Final step is to take intermediate D and remove its rejecting group, as seen here, by the use of a catalyst. Think about a catalyst converter and hydrogen gas. We have yet to perform the final step, but we have analyzed an asymmetrical intermediate compound D using proton NMR spectroscopy. Nuclear magnetic resonance, abbreviated NMR, aids in molecular identification and structural analysis. Hydrogen and carbon are dominant elements of organic compounds, so we typically focus on the NMR spectra when characterizing new compounds. The example shown here is an NMR for hydrogen, which we call proton NMR, and provides us information about the hydrogens in the compound, what they look like, what other hydrogens are close by, and how many of each one we have. For our asymmetrical compound, there are exactly 20 aromatic hydrogens, and these hydrogens typically show up around 7 to 9 ppm on our scale. If you look at the spectrum, you can see that integration values add up to exactly 20 as expected. Also in our compound, there are 8 methylene hydrogens, and due to these hydrogens being near electron withdrawing elements like oxygen or sulfur, and electron withdrawing groups like a benzene ring, we expect them to show up somewhere around 2 to 5 ppm on our scale. Looking at the spectrum once again, we can see that integration values add to nearly 8 as expected. In order to conclude that we successfully isolated a compound, we need to further analyze the compound using techniques such as carbon NMR spectroscopy, which instead of hydrogen tells information about the carbons within our compound, and liquid chromatography mass spectroscopy to confirm molecular mass or how big our compound is. In the future, we plan to perform our final reduction step to obtain our final desired product. Also, we plan to continue to explore more efficient, high-yielding synthetic routes for our monomers as our reactions are in equilibrium. I'd like to give special thanks to my research mentor, Dr. Nathaniel Schenk, for allowing me to take part in his research and for his kind and proper guidance. Also, I'd like to thank Georgia Southern Chemistry Department. Thank you for your time. I look forward to having concludes my presentation, and I would like to give special thanks to my research mentor, Dr. Nathaniel Schenk, for allowing me to take part in his research and for his kind and proper guidance. Also, I'd like to give thanks to the Georgia Southern Chemistry Department. Thank you for your time, and I look forward to answering any questions you may have. Thank you.